Hello, everybody. Today we are going to continue our discussion of female reproductive anatomy, physiology, and endocrinology by talking about the anatomy, physiology, and endocrinology of pregnancy, childbirth, and lactation. This is going to be the last full content uh, class for the semester. On Thursday, we will have a review class, and that will give you an opportunity to ask more questions leading up to the exam, the section exam, exam 3, on Tuesday, a week from today. So today we're going to talk about pregnancy, and then Thursday we'll have a review session, and then next Tuesday will be the exam. Please be sure to contact me if you have any questions or you need to seek adjustment about the exam schedule. So we're going to talk about ovulation and then fertilization of the oocyte by sperm. We will talk about the implantation of a developing embryo into the uterine endometrium. There is going to be 3 important hormones associated with implantation and pregnancy: chorionic gonadotropin, sometimes called human chorionic gonadotropin, or HCG. Estrogen and progesterone are all going to be important during pregnancy. Then at the end of the pregnancy, we will talk about labor and delivery briefly and talk about the hormones associated with labor and delivery: oxytocin from the posterior pituitary and prostaglandins from the uterus itself. And then we will turn our attention to milk, the production of milk by the mammary glands in a process that is called lactation, and the hormones that are important for the production of milk are, again, estrogen, progesterone, oxytocin, and then we will reintroduce dopamine and the anterior pituitary hormone prolactin produced by the lactotrophs of the anterior pituitary. So that's where we're going today. Feel free, be sure to ask me questions on the forum on Moodle. You'll remember this slide from last week. This is where we were at. So this cycle starts with ovulation, just like the previous one did. Here is a image of a oocyte surrounded by a corona radiata, which are granulosa cells, and the zona pellucida. So the corona radiata, the zona pellucida, and the oocyte in the middle are ovulated from the ovary and then picked up by the fimbria of the fallopian tube and transported by ciliary motility into the fallopian tube. The oocyte is viable in the fallopian tube and in the uterus for about 24 hours. In most cases of fertilization, the egg is fertilized by sperm in the fallopian tube. So the fallopian tube is the location for almost all fertilization events. So let's talk about fertilization a little bit more. There are obstacles to the sperm. So the sperm is introduced into the vagina by the male. There are obstacles to the sperm. The environment of the vagina is acidic, and this is not hospitable to the sperm, so it kills some of them. There is a plug in the cervix of mucus. Mucus plugs the cervix, and it serves to prevent sperm from entering the uterus. And if a sperm can't enter the uterus, it can't get to the fallopian tube, and so it couldn't fertilize. At ovulation, actually the estrogen levels that are increasing before ovulation cause a change in the mucus. And the mucus changes from sort of a thick mucus to a thinner, more liquid, stringier mucus that is more permeable to sperm. So just as ovulation is starting, the mucus plug to the cervix is changing and becoming more permissive for sperm to transit. There are also white blood cells, leukocytes, in the uterus that attack and destroy sperm, and they will actually proliferate once the blastocyst is implanted in the uterine endometrium. But they are present even before pregnancy. So about 300 million sperm are ejaculated. Half of them, if they get past the fallopian, get past the vagina, and they get past the mucus plug in the cervix, half of them are going to go up the wrong fallopian tube. And in the end, about 2,000 viable sperm reach the oocyte in the correct fallopian tube. So it takes them about 10 minutes from ejaculation to get up the fallopian tube and reach the egg if it is there, ready to be fertilized. But then it takes something like 10 hours of those sperms that get there, of those 2,000 sperms who get to the oocyte. It takes about 10 hours for them to fertilize the egg. And just as a final note, the sperm, even though the oocyte is viable for only about 1 day, the sperm are viable in the uterus and in the fallopian tube for several days. So the sperm can get there and before the oocyte is actually ovulated and transported into the fallopian tube. There is a process called capacitation of the sperm. Let me go through that for a moment. So here is a cartoon of a sperm with the head that contains the sperm genetic material. It's got the nucleus of the sperm. A midpiece that is rich in mitochondria and is basically a motor that spins the flagellum that causes propulsion of the sperm in the fluids of the vagina, the cervix, the uterus, and the fallopian tube. So the midpiece is where there are ATP-driven motors that spin the tail and drive like a propeller, drive the sperm to move. The plasma membrane of the sperm head is rich in cholesterol. So it's pretty sturdy. The vagina, the acidic environment of the vagina, tends to remove the cholesterol of the head of the sperm. So that process of removing the cholesterol, that sort of layer that protects the head of the sperm, is removed in the vagina. And then there is a layer called the acrosome, which is illustrated in yellow here. The acrosome is a layer inside the cell membrane that is now somewhat exposed because the cholesterol has been removed, and the acrosome is a rich environment of enzymes, and the enzymes of the sperm are going to become activated when they interact with the corona radiata of the ovulated oocyte. So when the capacitated sperm make contact with the corona radiata, enzymes from the sperm head are going to enable the sperm to penetrate the corona radiata and then bind to the surface membrane of the oocyte. This slide cartoons the process of fertilization. So up here is a cartoon of a oocyte surrounded by a zona pellucida, so itself surrounded by a corona radiata of granulosa cells. And coming in here are sperm. Here below to the right is a blowup of part of that oocyte with the zona pellucida, with the corona radiata and sperm approaching. Now there's something confusing about this slide. Once again, different points, different locations in space are actually representing time. So time is moving forward in this direction. So this is now time, not space. So here is a sperm that makes contact with the corona radiata, and the remember the corona radiata is formed from granulosa cells, and the acrosomal enzymes released from the sperm when it makes contact with the with the granulosa digest. They are proteolytic enzymes, and they digest the cells of the corona radiata. So then, but this sperm might not get all the way through, but it's helping to create spaces in the corona radiata where a lucky sperm, right here, this is the lucky sperm that gets through the corona radiata, and its acrosomal enzymes are starting to digest the zona pellucida. If it is lucky enough to get all the way through the zona pellucida, which is illustrated here, now the sperm has penetrated the zona pellucida and has fused with the surface membrane of the oocyte. The fusion of the sperm with the oocyte triggers an important event. That actually triggers several important events. First, it triggers granules that are located on the inner surface of the oocyte membrane. Those granules fuse with they undergo exocytosis. They fuse with the surface membrane, and that a wave of fusion, it's actually calcium driven, a wave of fusion of cortical granules with the surface membrane of the egg releases enzymes and causes the zona pellucida to now become impermeable to sperm. It cannot be digested by acrosomal enzymes, and so any other sperm, this is an unlucky sperm that got there too late and is repelled by the impermeable zona pellucida. So the sperm that got in and fused, its nucleus is released into the oocyte. That fusion, in addition to triggering the release of the cortical granules, also causes the nucleus of the oocyte to undergo the second meiotic division. So now we have two haploid genomes, the sperm haploid, the haploid genome inside the spermatid, and the haploid genome inside the oocyte. So this shows the process, instead of in a cartoon, it shows the process in a flow chart. Many sperm bind and acrosomal enzymes are released. Typically one sperm moves through the zona pellucida because its acrosomal enzymes were enough to get through the zona pellucida, and it binds to and fuses with the plasma membrane of the oocyte, and that causes important events. One is the sperm fuses and is drawn into the egg. The oocyte, the egg, completes the second meiotic division, and then the nuclei of the two haploid genomes fuse. In addition, the cortical granules, the secretory vesicles in this slide, but in the previous slide they were called cortical granules, fuse with the surface membrane. the enzymes from those cortical granules enter and change the zona pellucida and block any further sperm from entering. And the fusion also causes enzymes to be activated in the egg that leads to embryogenesis of the now fertilized oocyte. Here, let's start here. This is an image that shows fertilization and implantation. So here is an oocyte that was ejected or ovulated. It shows the oocyte on the inside, the zona pellucida in sort of pink color, and the corona radiata in the, well, I guess it's the corona radiata is not shown. The blue and pink are illustrating the zona pellucida in this cartoon. Fertilization that we have just walked through. The second meiotic division occurs. We have a sperm genome and an oocyte genome that fuse to form the zygote. This is illustrating mitosis, cell division, all that important stuff that you learned in CGI that I'm not going to review. Division of the fertilized zygote, leads to a 2-celled stage, and then a 4-cell, and then an 8-cell, and then 16 cells, etc. A rapid process of cell division. And the process of cell division develops a multicellular structure called a morula, which then develops into and when we get into the hundreds of cells, into something called a blastocyst. The blastocyst is what is going to implant, in the uterine endometrium and start to form the chorion, which will release chorionic gonadotropin. So this whole process, this development from one cell into a morula, into a blastocyst, and then into an implanted blastocyst, and then into the embryo, that developmental biology is a whole course, which I don't have time to go into. For our purposes, what we need to know is that a blastocyst implants in the uterine endometrium. The uterus has been prepared for this implantation. The estrogen and progesterone during the menstrual cycle that led up to the ovulation and fertilization helped to develop a thickened and enriched uterine endometrium. So progesterone from the corpus luteum continues to stimulate cell growth and swelling of the uterine endometrium. Remember, the corpus luteum is the source of progesterone, and it is maintained because chorionic gonadotropin was released in the process of implantation. So progesterone increases and stays elevated, and that helps to stimulate the proliferation and development of the uterine endometrium. The blastocyst continues to develop, and one of the important structures that it develops is the amnion, or amniotic sac. That's illustrated here. This is the amniotic sac surrounding a developing fetus. The amnion surrounds the embryo. It is filled with amniotic fluid, which is basically a filtrate of plasma from the maternal blood, as well as, especially in later stages, urine from the fetus itself. And the amniotic fluid and the amnion membrane protects the embryo and the fetus from trauma, from infection if there's any bacteria around, and it buffers against any temperature changes that might happen, although the maternal uterus does a lot of that too. The implanted the implanted blastocyst develops and causes many changes. And let's talk about the early development of the placenta. So let's get oriented in this slide. Out here is maternal blood flow in the uterus. Here is fetal blood flow in the amnion. This area here is supposed to illustrate the uterine endometrium, rich in blood flow, blood supply. These tree-like structures here are the villi, singular is villus, plural is villi, the villi of the chorion. So these are chorionic villi that develop and make contact with the placenta, well, really with placental pools of blood. So maternal blood forms these fluid-filled, blood-filled pools into which the fetal chorion are bathed. And so this is the source of exchange of nutrients, oxygen, and glucose and other nutrients from maternal blood into fetal blood, and the exchange of fetal waste products, carbon dioxide and other waste products into maternal blood happen at this contact here between the chorionic villi and maternal blood. The chorionic villi, there's how you spell it, the chorionic villi are the source of chorionic gonadotropin. And so these structures are important for nutrient exchange with the maternal blood. Implantation of the blastocyst and early development of the chorion, the chorionic villi, lead to the production and release of chorionic gonadotropin. So the level of chorionic gonadotropin in maternal blood increases dramatically at the beginning, and then it falls to a low, stable, but non-zero level throughout the rest of pregnancy. It is this spike in chorionic gonadotropin and the sustained high level of chorionic gonadotropin that sustains the corpus luteum throughout pregnancy. And you'll recall that the corpus luteum produces progesterone and estrogen, so that progesterone and estrogen levels continue to rise and stay elevated throughout the 9 months of pregnancy. At delivery, the levels of all three of these hormones, progesterone, estrogen, and human and chorionic gonadotropin, are going to fall dramatically. By the way, you may already know this, but it is human chorionic gonadotropin that is the hormone that is detected in the pregnancy tests. So those little sticks that you bathe in urine are engineered so that if there is chorionic gonadotropin present in the urine, the maternal urine, then the stick reveals a color change. And if there is no detectable chorionic gonadotropin, a different color change results as part of a positive control. In any case, it is human chorionic gonadotropin that is the basis for the blood test for pregnancy. I mean, the urine test for pregnancy. So during pregnancy, obviously there is all kinds of development. I mean, amazing process of development from a single cell into a functioning human being, all amazing development that we simply don't have time to cover. But let's cover what happens to really to the uterus during the last month of pregnancy. So beginning at around 30 weeks of pregnancy, a normal pregnancy is 40 weeks in length. So about 3/4 of the way through a pregnancy, there are weak and infrequent contractions of the uterine myometrium, the smooth muscle surrounding of the uterus. And typically these are so weak that they aren't even felt by the pregnant mother. But it starts at around 30 weeks and then continues to develop and increase in intensity. These contractions are a result of really a hormonal as well as a structural process in the smooth muscle of the uterus. Oxytocin, which is released in small amounts from the posterior pituitary, triggers contraction. Prostaglandins from the uterus itself in a paracrine signaling mechanism also triggers contraction of the smooth muscle. The smooth muscle cells of the uterus are all connected to their neighbors by intercellular channels, ion channels called connexins. So each smooth muscle cell is an individual cell, but it is coupled to its neighboring cells by gap junctions also, and the gap junctions contain channels called connexins. So contraction of one smooth muscle cell triggered by the mechanisms that you learned about in when we talked about skeletal muscle, a similar process of contraction in one smooth muscle cell will trigger contractions in its neighbors because depolarization of one smooth muscle cell can spread into its neighbors, triggering depolarization of those cells, triggering calcium release, calcium interaction with the contractile machinery, and contraction of the smooth muscle. So this process is starting at about 30 weeks, but it is weak, infrequent, and hardly detected. But as we get further along in pregnancy, the fetus shifts. Its head makes contact with the cervix. The fetal head makes contact with the cervix. And the cervix is stretched, and the collagen of the cervix begins to disintegrate. So contact of the fetal head and stretching of the cervix is part of the mechanical properties. So here's a flow chart. Let's start here. We have the head stretching the cervix. This stretch of the cervix causes a positive feedback loop. So there are signals from the cervix to the brain that cause the release of oxytocin from the posterior pituitary, and the oxytocin stimulates the contraction of the uterus. Stretching of the uterus and contraction of the uterus also causes it to release prostaglandins in a paracrine signaling mechanism that also stimulates uterine contraction. So we have 2 or maybe 3-ish stretch of the cervix also directly causes uterine contraction. So we have 3 very slow moving positive feedback cycles where increasing cervical stretch leads to increasing uterine contraction, and increasing uterine contraction then continues to signal and cause the release of oxytocin, prostaglandins, and physical stretch to trigger more and more uterine contraction until we get to labor. At a certain point, the contractions are strong, the amniotic membrane ruptures, and that really signals for the uterus to start very strong cervical contractions in the process of labor. Over the course of the next several hours, the uterine contractions increase in strength. The pressure of the baby's head on the cervix causes the cervix to dilate, and eventually this, if everything goes well, the cervix dilates, the contraction of the uterine myometrium pushes the baby through the cervix into the vagina, and the baby is born. Following delivery, the umbilical cord and the placental and the placenta, really the blood vessels in the placenta constrict. Blood flow to the baby from the in the placental blood flow really ceases. The placenta separates from the uterus and is delivered after childbirth as the afterbirth. Now let's turn to the process of production and release of milk, that is to say, lactation. During pregnancy, the high levels of estrogen leads to proliferation and development of the mammary gland itself. So the mammary gland, over here is a cartoon of the mammary gland, of the female breast and the mammary gland. Inside the mammary gland, inside the breast, there's layers of fat, and inside there are mammary glands. There are ducts that exist prior to pregnancy. The estrogen, the high sustained levels of estrogen during pregnancy, cause development of these ducts of the mammary gland. So prior to pregnancy, few ducts contain the so-called secretory alveoli. So let's jump ahead to progesterone. Progesterone, also sustained and high during pregnancy, causes the growth and development of secretory alveoli of the mammary gland. So prior to pregnancy, there are ducts. Let me see if I can clean this up. Prior to pregnancy, there are ducts in the mammary gland, but there is poor development of the alveoli, these bubbles of cells at the end of the ducts. So estrogen helps the mammary glands develop the ducts. Progesterone triggers those ducts to develop secretory alveoli, well-developed secretory sacs at the end of each of these ducts. So during pregnancy, the alveoli grow and develop, and they develop a lumen, that is to say, a fluid-filled compartment, which is where milk is going to be secreted and stored. The production of milk by the now well-developed secretory alveoli of the mammary gland is triggered by prolactin. So you will recall that prior to this, prolactin was under negative control of dopamine from the hypothalamus. Dopamine from the hypothalamus, released at the median eminence, transported to the anterior pituitary by the portal blood flow. That dopamine inhibited the lactotrophs of the anterior pituitary from producing prolactin. But the process, so prolactin was kept under negative control by dopamine. But the process of breastfeeding causes a decrease in the production of dopamine by the hypothalamus. Decreased dopamine then causes increased prolactin production. And the production of prolactin then triggers the well-developed secretory alveoli to produce and store milk. So it's the process of breastfeeding the process by which the baby stimulating the nipple of the woman, the breastfeeding is an important part of the production of milk. Mechanosensation of the nipple then triggers input to the hypothalamus. That the smell of the baby, the look of the baby, and the physical stimulation of the baby at the breast triggers the brain to tell the hypothalamus to inhibit dopamine secretion. So dopamine levels are inhibited, dopamine release is inhibited by this neural input to the hypothalamus. The decrease in dopamine then causes stimulation of the lactotrophs of the anterior pituitary to produce prolactin. Prolactin, interacting with prolactin receptors on the mammary gland, stimulates the synthesis and production of milk. The neural input to the brain of the woman also stimulates the posterior pituitary to produce more oxytocin. So the smell of the baby, the sight of the baby, the sensation of the baby increases the release of oxytocin into the blood. This oxytocin triggers another cell type that I haven't introduced yet, but the mammary gland, in addition to having duct cells and secretory alveoli, also contains myoepithelial cells, muscle-like, not muscle cells, but muscle-like cells that contract in response to oxytocin. So oxytocin triggers the contraction of myoepithelial cells in the woman's breast and actually causes the milk to be ejected, squeezed out of the nipple. So the stimulation by the baby, the suckling by the baby, I mean that sucks the milk out of the secretory alveoli, out of the mammary gland, but the myoepithelial cells also push the milk out in response to that suckling. So over here in text, suckling is what stimulates oxytocin release, and it inhibits dopamine release. It also inhibits GnRH release. So that's this process of breastfeeding, by inhibiting GnRH release, keeps LH and FSH levels low and is what prevents ovulation during the period of lactation following childbirth. So a lactating woman who is actively breastfeeding is not very fertile to another pregnancy because of the inhibition of GnRH release by the gonadotrophs of the anterior pituitary during suckling and breastfeeding. The oxytocin triggers milk ejection. The prolactin stimulates milk production. So prolactin is important for production. Oxytocin is important for milk ejection. And GnRH and LH and FSH levels are all kept low during breastfeeding. Now we could talk about some of the physiological changes in the mother's body during pregnancy. Most of the material that's going to follow here requires knowledge from ANP2. So some of you have this information, but many of you do not. And so I'm going to go through this without really any detail. And this will not be on the exam. So for example, the digestive system is activated during pregnancy, and the metabolic rate of the woman is activated during pregnancy due to thyrotropin, that is to say TSH, and increased thyroid hormone levels during pregnancy increase the metabolism of the woman and the increased absorbance of nutrients by the digestive system. So in AMP2, we talk about the digestive system, and now you know that this system is activated by pregnancy in order to increase metabolism during pregnancy, which is, the reason is obvious. The mother is developing a lot of cell growth and development. The circulatory system is covered in AMP2, and the main point here is that the output of the heart's cardiac output is increased by about 1/3 during pregnancy due to increased contractile strength and increased blood volume. There are changes in the respiratory system. So there is an increased demand for oxygen because of all of that metabolism and the need for more ATP by oxidative phosphorylation. So there is increased respiration, and there's a problem because the expanding uterus and the developing fetus actually impairs the ability of the lungs to expand, and so it can cause a process of apparent hyperventilation with a woman breathing rapidly and shallowly because the woman has decreased lung capacity, lung volume. There are changes in the renal system, the urinary system. Fluid is retained, so aldosterone, this is something that you know about, aldosterone levels increase, causing increased retention of salt and water during pregnancy. The filtration rate by the kidney, this is a concept covered in AMP2, that increases, and so the retention of fluid increases and the filtration of fluid increases. The expanding uterus and the fetus also presses against the bladder and reduces its ability to store urine. And so pregnant women often have to void urine more frequently than they were used to before pregnancy. Here is a cartoon of a developing baby fetus inside the myometrium, the well-developed uterine myometrium, and you can see how much the lungs are being impacted, how much the bladder is being impacted, the digestive, the bowel, the large and small intestines are being pushed aside as the baby grows and develops. So that's really where we're going to stop here. Again, the development of the fetus and the baby is an amazing process of developmental biology that we're not covering, but this did lead to the development of the baby and the process of childbirth and lactation, talking about the production of the first nutrition of the baby by maternal milk. Once again, please use the forum to ask questions. Ordinarily, we would have a class where you could bring your questions to class on Thursday, but instead I will prepare a PowerPoint that reviews some, but maybe not all, of the key concepts in order to refresh your memory. But using the forum is your opportunity for asking questions for this exam. You can send me email too, and if your question seems like a good question for everybody, I will post it to the forum. So please use email or the forum to ask questions leading up to the exam, which is on next Tuesday. And I will post information about the administration of that exam on Moodle.